Hi, so today I'm going to be presenting my biotechnology topic, DNA barcoding. What is it, how is it done, and what do we use it for? So DNA barcoding is a tool for rapid species identification based on DNA sequences. Basically what this means is it's a tool for identifying species very quickly based off of a small amount of DNA. And it's also used to speed up the process of discovering new species. DNA barcoding is revolutionary for many scientists, law enforcement, environmental conservation, and it was originally introduced by Paul Hebert, and he is a molecular biologist at the Biodiversity Institute of Ontario at the University of Guelph in Canada. So then we have a little bit more about him on the next slide. Like I said, his name is Paul Hebert. He is a molecular biologist, and he wrote his original paper on DNA barcoding in 2003. This paper proposed this large-scale idea that he could distinguish different species using a same small piece of DNA for every organism. Now, some 15 years later, DNA barcoding is used by tons of organizations with a ton of different species. They have a huge database for that now. Hebert says that his goal is to barcode every single organism on the planet. So now we're going to talk about how is it done. So basically, DNA barcoding is done by isolating a small sample of DNA from the subject. And this can be done, just picking up pieces of hair, pieces of poop, or actually extracting the DNA yourself. And then you amplify the sample using PCR, the polymerase chain reaction. And after you are finished amplifying the sample and you have lots of DNA to work with, you're going to sequence the PCR genes. So after that, it's going to be easier to make the barcode, read the barcode, and compare it to other genes in the database. And you have to do that to find the species that it is. You have to match the species to an already existing barcode. And then there's going to be several applications for DNA barcoding. Mainly it has the most promise in environmental control and conservation, things like that. If you are trying to preserve natural resources, you can use DNA barcoding to monitor illegal trade or find, what materials are going to be natural resources, how to protect those. It's mainly used by law enforcement in that aspect. It's also used for protecting endangered species in a similar way. So DNA barcoding can be used by law enforcement and able to test different meats maybe that are being sold, different materials to try to protect those species and catch the people who are poaching and selling them. Also be used to control pests because you can identify these pests in any life stage, which you can then figure out what it is, figure out how to eliminate that, and you have healthier agriculture. So it can also be used to identify disease carriers or disease vectors, in which you can identify the species that are causing diseases to animals and humans in order to understand and cure them or eliminate the disease carrier, such as mosquitoes. And then it can also be used in identifying medicinal plants. So if you have that plant in the DNA database, one, you'll already know what it is, but you can also discover new medicinal plants using this method of DNA barcoding. And then the final reason that I listed on here is you can monitor water quality with DNA barcoding. So basically, you can study the organisms that are living in lakes, rivers, streams, bodies of water, and you can, one, measure their health, but two, measure what should be there in a healthy body of water. So it can be easier to identify these species that otherwise you might not have been able to identify, and you can see why they're there, how they're doing, and how the water quality that they're living in is. So in the real world, DNA barcoding is super useful in species conservation, and it's really nice to have such a quick way to identify so many different species, as long as you have them in the database. In conclusion, DNA barcoding is a system for fast and accurate species identification. which is making environmental conservation efforts way easier. As I've said before, it has so many applications in so many different fields, such as controlling agricultural pests, sustaining natural resources, protecting endangered species, monitoring water quality, preserving natural resources. I can go on and on. Considering this is a relatively new concept, it has gone super far, and it's amazing how many species we now have in the database. Even where we're at with DNA barcoding, it is still a fairly new practice, and we do have a long way to go. However, it looks very promising from the amount of information that we have on it right now and how far it has come in such a short amount of time. Anyways, that is my presentation on DNA barcoding. What is it? How is it done? What is it used for? And I hope you all enjoyed. Thank you.